In this video, we will discuss about classification of PDAC and TME tissues using single-cell RNA-seq data. So in this experiment, they have used 3 PDAC tissue samples and 3 healthy persons tissue samples. So we are discussing about PDAC, pancreatic cancer cells from the previous video, where we have discussed that this paper has finally made a point that PDA's intracellular transport through exosomes in the PDAC will cause its lipid metabolisms, and if those intercellular transport will be disrupted, then it will disrupt the functioning of the pancreatic cancer cells, and it might even trigger apoptosis. So in this attempt, the paper begins with this classification of the 3 PDAC pancreatic cancer affected tissue samples, and along with that they also use 3 healthy tissue samples. And when they did this single cell RNA-seq data of this 6 samples, and after some filtering out and normalizations, they analyzed the top 2000 variable genes that are presented in the panel A. So this red one is the top 2000 variable genes. They have analyzed those genes, but this non-variable counts, they have not analyzed them further in their analysis. And in the PCA plot, they can see that this variable genes shows a linear property in almost all the samples, where so this indicates that these 2000 genes are highly variable among the samples, or within the samples there are variables. Even within the samples there is a high variability that can be seen here. And after that, what they did is they did this volume analysis, and in this case also they can find out that these 3 pancreatic cancer cells vary quite differently compared to the case of this normal tissue samples. The normal tissue samples has almost similar variations compared to the case of this pancreatic cancer cells. So this indicates that this violin plot indicates that the cancer cells not behave uniformly, but behave differently in different patients. And these things they have validated using the standard deviations of this pancreatic cancer samples, and they can see that this standard deviation varies across the pancreatic cancer samples, across the pancreatic cancers, that can be seen in this panel C. After that, they did the clustering of this or they did the Tisney plot to reduce the dimensionality of the principal component analysis. And in this case they are able to see different clusters of the cells are formed, and they differentiate the tumor cells from the healthy cells using different colors that can be seen here. The light green represents the normal cells, and this dark blue represents the tumor cells. After that, they did the literature analysis to find out the markers for each cells, find out the markers for each cell lineage, and based on that literature they differentiate they categorize these clusters into two different group, not two different group, into this 19 into the 20 different clusters, and they categorize these 20 different clusters both in the normal tissues and also in the tumor tissues. And after that they decided to find out which of them are more prominent in these samples, and they can see that around some of the cells they are able to find which are more prominent, that are mentioned here. For example, in this cluster 18, they are able to see that TTR is more prominent, and this one is like in the cluster 19, they can see that TPSB is more prominent. So this is the analysis that they did from the literature review, that these gene markers are more prominent in this. So based on that, they characterize this cell populations as the belongs to the cell lineage where these gene TPSAB1 G markers are present. So they consider them as those cell lineage which are dominant in this TPSAB1 genes. So in this panel E, they just characterize both the normal tissues and normal samples and the tumor samples into different clusters based on their Disney plot. And in the case of this panel F, they use the literature review to find out these clusters belongs to which cell lineage. In the panel G, what they did is they characterize them based on the literature reviews and the gene markers that are present in those samples, in those clusters. And after that, they compares that in the case of tumor cells, they see the decrease in this acinar cells compared to the normal cells. However, they see the increase in the epithelial cells in the tumor cells compared to the normal cells. And a few things are also decreased in the tumor cells, such as macrophages are decreased, and also these endothelial cells are decreased in the tumor cells. So these are some of the major changes that they have identified so far from this single cell RNA-seq data, indicating that there is a cell composition changes occurs in the tumor cells along with this tumor microenvironment compared to the normal cells. So this is what the main outcome from this figure, from the experiment, these experiments. And if you need to understand more about their analysis techniques, how they have filtered out the other genes and how they come up with this 2000 variable genes, you can go through the all the other analysis. You can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.